Welcome to SWAM NPTEL course on food microbiology for safe and sustainable food systems. We have been discussing mechanism of microorganism control in food, and in that we have discussed so far there is thermal processing of food. In that we have discussed blanching, pasteurization and appertization. After that we have discussed sterilization, then thermal death rate kinetics, irradiation, and now we will be discussing in this lecture the chemical methods for microbial control in foods. And the topics which we will be covering will include introduction, high level germicides, intermediate level disinfectants and antiseptics, low level disinfectants and antimicrobial agents. Now, chemical agents for the control of microbial growth are either microcidal, microbiocidal or microbiostatic. Microbiocidal agents are stringent. Microbiocidal agents are sterilant which kill all living cells. Microbiostatic agents kill some cells and inhibit the growth of others. The spectrum of activity exhibited by any microbiocidal or microbiostatic agent is an important factor in choice and should be considered along with potential harmful effects on the user. An agent which kills staphylococci may be totally ineffective against mycobacteria and would be useless in a tuberculosis ward. There are four large categories for agents of chemical control. Antibiotics are produced by microorganisms to kill or inhibit the growth of other microbes. These agents are generally selectively toxic and can be naturally produced, synthesized or semi-synthetic. Antiseptics are synthetic compounds which kill or inhibit the growth of microbes in the surface of the skin. Disinfectants are chemical compounds which kill or inhibit microbes on the surface of fomites. Preservatives such as sugars, salt, nitrates, nitrites, sulfate and sulfites inhibit microbial growth in food, usually by producing osmotic environments which are unfavorable to microbial growth. These can be further subdivided as high, intermediate or low level agents. High level germicides sterilize fomites but are toxic to skin and mucous membranes. Intermediate level disinfectants and antiseptics kill and inhibit on fomites and skin but can be toxic to user at medium to high concentrations. Examples include phenolics and halogens. Whereas low level disinfectants such as alcohols, hydrogen peroxide, heavy metals and soaps kill some microbes but inhibit the growth of most. An ideal antiseptic or disinfectant should have following properties, like it should have wide spectrum of activity, it should be able to destroy microbes within practical period of time, it should be active in the presence of organic matter, it should make effective contact and the wettable, It should be active in any pH. It should be stable. It should have long shelf life. It should be speedy, should have high penetrating power, should be non-toxic, non-allergenic, non-irritative or non-corrosive. It should not have bad odor, should not leave non-volatile residue or stain. Efficacy should not be lost on reasonable dilution, and lastly it should not be expensive and must be available easily. Now, high level germicides. These are called agents of cold sterilization, since no heat needs to be applied to increase their activity. These are generally alkylating agents which kill by adding ethyl or methyl groups to nucleic acids or proteins. While the agents are capable of killing vegetative cells, spores and inactivating viruses, some take up to several hours to complete their germicidal activity. Aldehydes such as formaldehyde and glutaraldehyde fix tissues by alkylating and forming cross links between adjacent proteins. They are commonly used as fixative compounds for electron microscopy, preservatives of specimens and cadavers in some synthetic plastic compounds, and can be used to sterilize anesthesia tubing and surgical implements. Aldehydes can fix living tissues such as mucous membranes and have the ability to vaporize or outgas from compounds containing them. So, they should be handled with caution. Beta propiolactone is a liquid alkylating sterilant with a high boiling point of 155 degrees Celsius. It is generally used to sterilize bones used in grafts. It quickly breaks down into non-toxic compounds when it comes into contact with organic matter, but can burn skin. Ethylene oxide or carboxide kills vegetative cells and spores. It is a liquid at temperatures below 10.8 degrees Celsius, but rapidly sublimates into a highly inflammable gaseous state above this temperature. It is generally used in a chamber similar to an autoclave at 60 degrees Celsius for 1 to 10 hours, where it is mixed in a 9:1 ratio with carbon dioxide, that is 90% carbon dioxide, 10% ethylene oxide, which reduces its toxicity but also its inflammability. Carbon dioxide carboxide can be used to sterilize surgical implements and glassware, but these fomites must be allowed to degas before use, since residues can stimulate mutations in bacteria. Ozone occurs naturally in the upper atmosphere, where it serves to shield the surface of the earth from solar radiation and is produced as an exhaust gas by vehicles and industry, acting as a pollutant in the lower atmosphere. Applied properly in a chamber, ozone is a powerful oxidizing agent which kills cells and spores on the surface of glassware, surgical implements and bandages. An advantage of sterilization with this compound is that it outgasses quickly, leaving no toxic residues as can ethylene oxide and beta propiolactone. Now, intermediate level disinfectants and antiseptics. Phenol, that is carbolic acid, is one of the earliest disinfectant compounds to be used in healthcare facilities and laboratories. Phenol kills microorganisms by denaturing proteins and destabilizing cell membranes, is bactericidal, fungicidal and virucidal at high concentrations, but is not effective against bacterial endospores and is effective against many potential pathogens including mycobacteria, Staphylococci, Streptococci and gram-negative coliforms such as E. coli. It can be used to disinfect garbage cans, surgical operating facilities, laboratory equipment, feces, urine and sputum, but it is very corrosive at higher concentrations and its fumes can be lethal. Because of its toxicity, this compound is generally used as a solution between 2% to 5% in concentration, and many fewer toxic derivatives have been produced. The most common of the phenolic derivatives are called cresols and bisphenols. Cresols are formed by adding methyl groups to phenol and are used for the prevention of wood products as compounds such as creosote, that is paracresol. Bisphenols are produced by combining two phenol molecules. Halogens are a family of elements with a high affinity for electrons. This affinity makes them very reactive with biological molecules, and they can serve to disrupt enzyme activity, break down lipid structure and produce oxidizing agents such as singlet oxygen or nascent oxygen. The halogens most commonly used as disinfectants are chlorine and iodine. Chlorine is used as a disinfectant only, either as a gas or in liquid form, which is effective against many vegetative forms of microbes as well as some viruses such as hepatitis. Iodine is lethal to all vegetative forms of microorganisms, can inactivate viruses and is fairly effective in higher concentrations against endospores. Pure iodine is caustic to tissues, so it is diluted with other compounds. Tincture of iodine is produced by dissolving crystalline iodine in alcohol. Now, low level disinfectants. Hydrogen peroxide is a good low level disinfectant agent when used in concentrations of 3% or lower. Higher concentrations are caustic to human skin and cannot be used. Obligate anaerobic microbes are especially sensitive to hydrogen peroxide since they do not produce catalase and the rapid release of oxygen gas inhibits their growth. Alcohols such as ethanol and isopropanol are effective antiseptics and disinfectants when used in concentrations between 70% and 80%. Alcohols kill microbes by denaturing proteins, dehydrating at 100% concentration, and as nonpolar solvents which disrupt the phospholipid structure of the cell membrane but are relatively ineffective against spores and viruses. Also, dehydration may actually be beneficial to some microbes, enhancing their survival by extracting extracellular water. So, alcohols such as ethanol are normally used in lower concentrations. Some dyes can not only be used to stain microorganisms, but also have antimicrobial activity. Crystal violet, which is used in very low concentrations as gentian violet, can be used to treat oral infections by bacteria such as Rockleynia quintana, the agent of trench mouth, and fungal infections such as Candida albicans, which causes oral thrush. Now, antimicrobial agents. Antimicrobial agents are used for treatment of diseases by killing or inhibiting the growth of susceptible microorganisms. These can be chemically synthesized, that is chemotherapeutic agents, or microbial products or their derivatives, that is antibiotics. These agents generally act by inhibiting one of the metabolic processes of the microbe or block the synthesis of important component such as cell wall. A good antimicrobial agent is expected to have selective toxicity. It must kill or inhibit the pathogen microbe without having any or minimum harmful effect to the host. Selective toxicity can be measured if we know about the toxic dose and therapeutic dose of a drug. Toxic dose is the level of the agent or drug at which it becomes toxic for the host. and therapeutic dose is the level of the agent or drug required for the treatment of the infection. A ratio of toxic dose to the therapeutic dose is known as therapeutic index. So, higher the therapeutic index, better is an antimicrobial agent. A drug that disrupts a microbial function not present in the host has greater selective toxicity and higher therapeutic index. For example, penicillin inhibits the synthesis of bacterial cell wall, has little effect on the host as it does not contain cell wall, making it a drug of high therapeutic index. If a drug acts on a pathway which is common in both host and pathogen or disrupts some other pathway in the host, the outcome is undesirable in the form of side effects and the drug is said to have low therapeutic index. The range of effectiveness of antimicrobial drug varies. They may be narrow spectrum drugs that is effective only against a limited number of pathogens, or broad spectrum drugs which is effective against many pathogenic microbes. However, based on microorganism, they are classified as antibacterial, which is active against bacteria, antifungal, which is active against fungi, antiprotozoal is active against protozoa, and antiviral, which is active against viruses. Now, mechanism of action of antimicrobial drug. First is inhibition in synthesis of bacterial cell wall. Penicillin stops the synthesis of cell wall of gram-positive bacteria that are synthesizing new peptidoglycan. It inhibits transpeptidase enzyme which are responsible for cross-linking of polypeptide chain of the bacterial cell wall peptidoglycan. It also activates cell lytic enzymes. Second is disruption of cell wall. Polymyxin B binds to microbial plasma membrane and disrupt its structure and permeability. Now, alteration in membrane function. Gramicidin can alter the function of cell membrane. It brings about a change in permeability of cell membranes and quick loss of amino acids, minerals, phosphorus, and nucleotides. The inhibition of protein synthesis. Streptomycin binds to 30S subunit of ribosomes and causes misreading of mRNA and therefore inhibits protein synthesis at the final stage, that is during transport of amino acetyl tRNA to ribosomes. Inhibition of synthesis of purines and pyrimidines. Mitomycin C inhibits the growth of many bacteria, protozoa, and arrests the growth of tumor cells. The chromatophore of actinomycin is incorporated between DNA base pairs, whereas mitomycin blocks the synthesis of DNA without affecting RNA and protein synthesis. Inhibition of respiration. Antimycin inhibits growth of some fungi. It inhibits oxidation of succinate. Valinomycin is active against gram-positive bacteria by inhibiting oxidative phosphorylation. Now, antagonism of metabolic pathway. There are antibiotics which block the functioning of metabolic pathway through competitive inhibition of key enzymes. Such valuable drugs are known as antimetabolites. An example is sulfonamides. Sulfonamides inhibit folic acid metabolism by competing with para-aminobenzoic acid.